We've been talking about the brain as this energetically efficient device that just uses 15 watts of power. What I want to do in this lecture is talk about that 15 watts, talk about what it is that that 15 watts of power is buying us. And what we're going to do is we're going to talk about what the energy is being spent for. And by the end of this lecture, what we're going to see is the answer. The answer is that the 15 watts of power is used to power an elaborate chemical mechanism that generates the signals of the brain. Now, often I'm asked the question by students and also by non-scientists, is the brain chemical or electrical? And I think that when people ask this question, they may be thinking of an analogy to a computer, in which electrical signals, which after all are what the brain generates, they're thinking about computers in which electrical signals are carried by electrons and wire. Now, it is true that the brain generates electrical signals, but fundamentally, the brain is chemical in nature, and it's chemical in its nature in two different ways. The first way is that the electrical signals of the brain are directly generated by chemicals. These chemicals are called ions. So for instance, when you dissolve salt in water, it comes apart into ions, and those ions are what your tongue detects when you taste salty solutions. So cells move ions around, and the movement of ions across the membrane of a cell are the events that allow the possibility of generating electrical signals. And so that's one way in which electrical signaling in neurons is fundamentally chemical. There's another way in which neurons and brains are chemical in nature, and that's that chemicals are also used as signaling molecules. They're used as signaling molecules to send information from one neuron to another neuron, and that's a process that's called synaptic transmission. And so neurons talk to each other through the release and detection of chemicals. And furthermore, these chemicals that neurons use are also used not only to talk from neuron to neuron, but also to send information inside cells. So for instance, a signal arriving at a neuron then can lead to the generation of a chemical inside the cell. And that information inside the cell can then be used to process information and even to store information and to generate changes that can last for a second up to a lifetime. And those chemical signals are a fundamental part of what brains do. Now, what we're going to do in this lecture is talk about how these signals are made. And so in order to understand how these signals are made, we're going to start by looking at neurons. So let's just think about what neurons are. Now here's an example of a neuron. It's a reasonably realistic looking neuron. Fundamentally, neurons of the brain are cells. Neurons and glia are cells, and that's what brains are made of. And so if you look at this neuron, this is a sort of a more schematic version of a neuron than what I showed you before, but perhaps it's a little bit easier to see what's going on. Let's see. So this is a neuron. This particular neuron is a motor neuron. So often we're going to be talking about neurons of the brain where neurons talk to other neurons. This particular neuron is a neuron. It's a cell, like all neurons, and it sends its axon, in this case, to a muscle fiber. So this strip here that looks kind of like a strip of bacon is actually a muscle fiber. And the idea here is that this neuron is sending its axon, this output structure, the axon, then goes and sends signals to the muscle. So in this case, it's a neuromuscular junction, but often neurons, of course, talk to other neurons in the brain. And most neurons of the brain talk to one another. And so what we have here is a neuron, its cell body, its axon, the electrical signal goes out on this axon and then innervates this muscle and causes the muscle to contract. Then neurons also have dendrites. Here's an example of a dendrite here. So this is a dendrite, and this dendrite is an input structure in which the neuron receives information, and it comes in on synapses. So the synapses are formed here on the neuron, and then those synaptic inputs then generate electrical responses that are picked up by the cell body and turn into signals that go out. Okay? So that's what a neuron is. So looking at this neuron, what I want you to think of when you look at this neuron is not all the particular cellular details. We've got a nucleus and all this other guts of the cell here. For the moment, let's think of the neuron as a watery bag of salts. This dendrite, these axons, all the bits of it, the cell body, this is all basically a bag filled with salty water. And this bag is a bag of salts where the inside has different concentrations of salts, of ions, compared with the outside. Now, the way that the concentrations of ions are different between the inside and the outside of the neuron is that ions cannot easily get across a plasma membrane. Ions can only be dissolved in water, and so as a result, ions cannot get across a membrane that's made of fat molecules alone. So a key player in how neurons work is that neurons are composed of this bag of salts surrounded by membrane, and the membrane is studded with proteins. These protein molecules sit in the membrane, and these proteins allow ions to move back and forth across the membrane. And this back and forth movement of ions across the membrane generates then electrical currents the way that a battery would. Now these electrical signals, these electrical currents, are the components of the electrical signals of neurons. In particular, I'm going to show you now how these electrical currents generate the fundamental currency of how neurons signal, the action potential. Now, it's possible to measure the voltage inside a neuron. As I've mentioned in a previous lecture, you can do so by sticking an electrode into the neuron. And when you stick an electrode into a neuron, what you see is a voltage of the inside of the cell compared with the outside that's a little bit different. And it's a little bit different because this membrane is a very poor conductor, and then the voltage inside the cell is different from the outside of the cell. And what happens when you put in the electrode is that the voltage sits at a voltage of around minus 70 millivolts, a little bit negative compared with the outside. If you inject a little bit of electrical current into the neuron, then the voltage changes a little bit, and then it comes back. And if you inject a little bit more current into the neuron, you get a little bit more voltage change. Now, something special happens if the change is large enough. If you inject enough current into the neuron, as an experimentalist, what you see is, boom, a change in voltage in which the voltage changes rapidly, goes up to above zero, and then comes back to rest. And this is the spike that we've been talking about. This spike is a fundamental unit of signaling in the brain. It's an all or none signal. Either a neuron spikes or it doesn't. And this fundamental currency is how neurons send signals over long distances, how neurons translate the inputs that they get into an output. Now, let's dig in a little bit here. How is this spike generated? Now, the way that neurons generate action potentials, the way that they generate spikes, has four components to the explanation. And let me just walk through it with you. So firstly, I told you that membranes are studded with proteins that let ions go back and forth. One major type of protein is ion channels. Ion channels are little pores made of protein, and these little pores made of protein can open and close. And what happens is that when they're closed, ions cannot get across the membrane. When they open, these pores act like little bridges. And these little bridges let molecules, let ions across. And when these bridges open up, it's like a toll bridge, and so when they open up, ions can cross in a direction from high to low. So I said before that ions are at different concentrations on one side of the membrane compared to the other. When these channels open, what happens is that ions flow from the side of high concentration to the side of low concentration. And these bridges often only let particular ions through. So for instance, you might have a channel that only lets sodium through, and that would be called a sodium channel. Or you might have a potassium channel, which only lets potassium through. So the first component of the explanation is that the membrane has ion channels that can open and close. Now the second component is what I said before, which is that the concentrations of ions on one side of the membrane and the other side of the membrane are not the same as one another. And what that means is that when these ion channels open, there's a movement of ions from the direction of high concentration to the direction of low concentration. Okay, so for instance, there's about 4 times as much sodium outside neurons than inside. And what happens at the beginning of an action potential is that sodium channels open. And so when sodium channels open, sodium rushes from the outside of the cell into the inside of the cell. And as a result, what happens is that these sodium ions, which are positively charged, go from the outside to the inside. As a result, positive current flows into the cell, and the voltage inside the cell goes up. Now next, another component of this is that there's more potassium inside neurons than outside. Potassium is another ion that's positive. So now you've got all these positive ions inside the cell, potassium ions, and there's a lot more inside than outside. And so during the next phase of the action potential, what happens is that potassium channels open, and they open a little bit more slowly. So they open after the sodium channels have opened. And so now what happens is potassium channels open, a little bit of positive charge flows out in the form of potassium ions, and now the voltage goes back down again. All right? And so what happens is the voltage goes up because of sodium entering the cell, and then it comes down because of potassium exiting the cell. Now the last component of this explanation is that these ion channels are themselves sensitive to voltage. And so they're affected by voltage. They alter the voltage because they open and allow charge to cross the membrane, and they also are sensitive to voltage. And this generates a phenomenon known as feedback. So think of examples of positive feedback from everyday life. An example of positive feedback is fire. So in fire, you have heat generated by the fire, and that heat generates vaporizes particles of wood and then leads the wood to combine with oxygen, and then therefore leads to more combustion, which then leads to more fire. And that's an example of positive feedback. Another example of positive feedback would be a microphone in an auditorium, perhaps where the sound system is turned up too loud. When the sound system is turned up too loud, then the sound goes into the auditorium, into the microphone, and then generates feedback and an unpleasant ringing sensation, right? So that's another example of positive feedback. So this positive feedback is a key component of how action potentials are generated. So to summarize what we have here as the biophysics of action potentials, the physical mechanism by which action potentials are generated is sodium comes in, then potassium comes out, and both of these processes are themselves part of a feedback loop in which voltage leads to further changes in voltage. And so that feedback mechanism is how action potentials are generated. Now, I've set up for you a situation in which sodium and potassium concentration differences are key to generating the action potential. And these concentration differences are called gradients. But how did these gradients get set up? How did this come to be in the first place? And this is where the 15 watts comes in. So the potassium and sodium concentration differences across the membrane are set up by other protein molecules in the membrane. So far I've talked to you about ion channels. There's another kind of protein that sits in the membrane, and it's called an ion pump. And these ion pumps use up energy and have the job of moving ions across the membrane. So when an ion channel opens, it is passive and just simply allows ions to cross the membrane in the direction of high to lower concentration. There's also another category of molecules called pumps. And the main pump that is operative in setting up neurons in their concentration gradients is called the sodium-potassium transporter. It's called the sodium pump for short. And what the sodium pump does is it sets the chemical stage. It basically takes sodium and transports it out of the cell. It takes potassium and transports it into the cell. And so it's constantly moving sodium ions out and potassium ions in. And that process is the process of stage setting to allow the concentration of ions across the membrane to be different. Now, because this pump is moving ions against their direction of natural movement, what this pump is doing is using energy. It costs energy to set up these gradients. And so this 15 watts that we've been talking about is mostly used in the form of, is mostly used to establish these chemical gradients by moving these ions across. And the energy comes from a molecule called ATP. ATP stands for adenosine trisphosphate. ATP is a molecule that is a source of energy. It's a universal source of energy in all cells. What happens is that ATP binds to the sodium pump, and when it binds to the sodium pump, then the pump moves 3 sodium ions out of the neuron and moves 2 potassium ions into the neuron, and in doing so, breaks a chemical bond in the ATP, and that's where the energy comes from. Okay, so that's where the energy comes from for setting the stage for neurons. Now, the ATP's chemical energy comes from another source. It comes from glucose, and we all know of glucose as sugar, a universal energy source for the body. Glucose is used as an energy source to generate ATP with the help of oxygen, and this is a biochemical process known as oxidative metabolism. And this requirement for oxygen is why the brain is so sensitive to oxygen deprivation. In fact, the brain is damaged after just a few minutes without oxygen. So to summarize, sodium pumps set the stage. They set the stage by building these ionic gradients, these concentration gradients that allow action potentials to be generated. And they also are constantly cleaning up. They're like janitors whose job it is to get rid of the sodium and potassium. And they're constantly cleaning up after all the spike activity and also other electrical cells that are generated in neurons. So these sodium pumps are central players that take hours and hours to constantly be moving sodium and potassium ions, constantly getting neurons ready to do the thing that they do when we engage in neural processing. Now, I said that spikes use feedback. I said that action potentials operate by a central principle of voltage feedback, and I used an analogy to fire, right? Fire leads to more fire. Now, this feedback property also allows spikes to travel over long distances, and this capacity to travel over long distances is the key to what makes it possible for messages to get across the brain. So let's start with the fire analogy. So I talked about fire as being just the stationary fire that has positive feedback and allows more fire to be generated. Now imagine a line of gunpowder. So when one part of the gunpowder blows up, it ignites the next part. And then that part of the gunpowder ignites, and then the next part. And that feedback process now allows the fire to travel over distance. Now something analogous to this happens in axons, and this is the axon, recall. And what happens in axons is that an action potential that's generated in one part of the neuron, in one part of the axon, then causes the voltage to change in the next part of the axon. And this feedback process now goes from one part of the axon to another and allows the action potential to spread from one location to another, and then it can travel down the axon towards where the axon is headed, towards other neurons, or in this case, a muscle fiber. This process is called conduction. Now, axon conduction travels at finite speeds. There is a limit to how fast signals can travel. It is in some ways limiting to how fast we can think. This conduction speed ranges from about 0.2 meter per second, which is about half a mile per hour. This is about half a mile per hour right here. Right? So that's the slowest that an action potential can travel. The fastest that an action potential can travel is about 100 meters per second, and I'm not going to demonstrate that for you, okay? But 100 meters per second is close to 250 miles per hour, which is why I'm not going to demonstrate it. That high speed occurs in wide axons, and so, for instance, the axons that lead from the brain down the spinal cord towards the fine finger musculature, that's an example of a pathway that contains very wide axons. And that's responsible for how it is that we're able to make coordinated movements at a very high speed with our fine finger movements. Now, these fast conducting axons achieve their speed partly by an insulating sheath called myelin. And if we go back to this, we can see that there's a sheath of myelin. It's indicated by this little orange guy here. Myelin is made by glia, and this is a glial cell. And what happens is that the glial cell makes this insulating sheath that then wraps around the axon and forms an insulating sheath that then makes it so that there's less charge that's necessary, less electrical charge that's necessary for this axon to then generate an action potential. And this insulating sheath allows the axon to be more efficient and therefore allows impulses to travel more quickly down the axon. Now, it turns out that axons are the only means of long-distance communication in the brain. So these axons are the only way that signals can travel over a distance. And as I mentioned, there are neurons in the brain that sends axons all the way down the spinal cord from the cortex down to control the muscles. And in the case of, for instance, whales, these axons can lead many meters, perhaps something like 10 meters in the largest of whales. Now, these axons that go this long distance are often very fast. As I mentioned, the ones that control the fine finger movements are very fast. But some are very slow. When I demonstrated the slow movement of axon conduction, if you think about how long that takes, it turns out that the very slowest impulses cross the brain in as long as 1 second. So evidently, some brain functions don't require speed, because we, in fact, have these very slow axons in our brains that cross from one side of the brain to another. So there's something going on there in which axons don't always have to be fast. Now, I've told you about axons. I've told you about how they are used to send messages over long distance. Let's think about where they are in the brain. So far, I've told you about the obvious place where they have to be in the brain, which is going from the brain down to the musculature. It turns out that axons are going from place to place within the brain. And if you look at brain tissue, you can see gray matter and white matter. The gray matter is composed of all kinds of structures. It's composed of neurons, glia, axons, but it's also composed of dendrites and synapses. And so there's all kinds of stuff in gray matter, and it ends up looking gray when you look at it, just if you cut up a preserved brain or if you look under a microscope. White matter is different. White matter is composed of only axons, and these axons are ensheathed in myelin, and the axons and myelin are somewhat fatty, and as a result, it's white looking, and that because it's made entirely of axons. Now, the white matter, therefore, is a place where there are highways from one part of the brain to another part of the brain. The white matter is where there are connections over long distances. The biggest highway is a superhighway. It's the superhighway of connections between the left half of the brain and the right half of the brain. And this highway, this superhighway, is called the corpus callosum. And if we look at a top view of the brain, we can see the corpus callosum as being this band of fibers that crosses from left to right, right to left. And this band in our brains contains billions of connections. This is how the halves of the brain are integrated. This is how the left half of the brain and the right half of the brain communicate with one another. And when this pathway is cut, then the halves of the brain can't communicate with each other. And this is how it was discovered that the halves of the brain, the left half of the brain and the right half of the brain, each have their own general abilities, each have specialized jobs. It turns out that the scientist Roger Sperry won the Nobel Prize for understanding this and for looking at patients who had the halves of their brain separated by a surgical operation to try to deal with intractable epilepsy. And the idea was that people had seizures on one half of their brain, and these seizures would spread. And neurosurgeons had the idea to cut at the midline and cut all these fibers of the corpus callosum and prevent signals from getting from one half of the brain to the other half of the brain. And there was an inadvertent outcome of this, which is by cutting the brain in half, what ended up happening was that the left half of the brain and the right half of the brain were no longer able to communicate. And in some sense, what those surgeons generated was two people for the price of one. So two halves of the brain not communicating with one another because of this operation and because the axons were cut. Now, so far we've gotten from the neuron down the axon. But then what? Information goes down the axon to talk to other neurons. At this point, action potentials cannot jump to the next neuron. What happens at this point is that the spike arrives at the end of the axon and it causes a chemical signal. Remember that I said that chemicals are used for neurons to talk to one another. The chemical signal that happens is that when the spike arrives at the end of the axon, it causes neurotransmitter to be secreted from the end of the axon. And the process of the secretion is synaptic transmission. The way this works is that neurotransmitters are released from the ends of axons. The spike arrives at the end of the axon, causes bags of neurotransmitter inside the cell. These bags are called vesicles. And these vesicles fuse with the membrane and then dump their contents into the space between neurons. And that dumping is called neurotransmitter release. That released neurotransmitter then diffuses across to the other side where the neurotransmitter binds to dendrites and specifically binds to receptors on the surface of dendrites. And then the receptors on the other side of the synapse, on the dendrites, are also ion channels. So far I've told you about voltage-gated ion channels like sodium channels and potassium channels. It turns out that neurotransmitter receptors are also ion channels. And these ion channels then bind to neurotransmitter and they open. They're like little bridges that allow ions to cross. And so it's the same story that I was telling you about about action potentials. And what happens is that when the neurotransmitter binds to the receptor, channels open and there's an electrical signal. And that electrical signal is called a synaptic potential. And that synaptic potential then can make a neuron more likely or less likely to fire. If it makes the neuron more likely to fire, that's called excitation. If it makes the neuron less likely to fire, that's called inhibition. And what happens when a neuron receives information is that the neuron receives information from thousands of other neurons, and those thousands of other neurons either excite or inhibit that neuron. And generally, one synapse, one connection from another neuron, is not enough to make the postsynaptic neuron spike. What happens is that many of these thousands of neurons come together, and if there's enough net excitation, if there's enough excitation and a sufficiently small amount of inhibition, that net excitation is sufficient to cause that neuron to spike. And so that process is referred to as synaptic summation. Now, we've talked about electrical signals of neurons. One kind is action potentials, these very fast signals. Those signals take about 1,000th of a second, 1 millisecond. I've also told you about synaptic potentials, these events that happen when one neuron talks to another neuron. Those take about 10 times longer. Typically, they take typically around 10 milliseconds. These signals together constitute the electrical activity that occurs inside brains. And it turns out that it's possible to measure this electrical activity. And a major method that experimentalists use and also clinicians use to measure brain activity is a technique called EEG. EEG stands for electroencephalography. And EEG is a method for measuring electrical activity. It's a method in which electrodes are put all over the scalp on the outside. And what EEG measures is the summed activity of all the neurons of the brain. And so each neuron and each synapse is generating these very small currents, which would be unmeasurable if you put the electrodes on the outside of the skull. But these 100 billion neurons and trillions of synapses working together then add up to make a signal that can be picked up by EEG. And so therefore, if these signals vary together, if neurons on average are firing together in sync, then that generates a total signal that can be measured. And this total signal is what EEG picks up. And it turns out that EEG recording largely picks up synaptic currents. And these synaptic currents are much of what is picked up by EEG recording. And so now I've explained to you how these electrical signals are the currency of brain communication and are also signals that can be picked up by making an EEG recording. Now let's summarize what we've learned today. We've basically learned how neurons make electrical signals. I've told you that the membrane is made of fat, and it's an insulator, but it has proteins in it that let ions across. And these proteins come in multiple categories. One category is ion pumps. Ion pumps set the stage for electrical signaling by moving ions across the membrane and creating concentration gradients. And then there are ion channels. Ion channels are little bridges that allow ions to cross passively, and that passive opening is a key component of generating electrical signals in the brain from moment to moment. A critical component of that signaling is the action potential, the spike. And this is an event in which there's an all or none signal that's composed of sodium entering the cell and potassium exiting the cell. The other major component of electrical signaling in brains is synaptic transmission. And in synaptic transmission, there are other ion channels. These ion channels are sensitive to neurotransmitters. And these neurotransmitter-gated ion channels that are opened by neurotransmitter, are how neurons communicate with one another. Now, it turns out that neurotransmitters don't just open ion channels. They also do other things. There are other receptors that play a critical role in neural signaling, and these other receptors for neurotransmitters are enzymes, not ion channels. And that turns out to be a major part of what brains do, and it turns out to be how drugs and other chemicals work upon the brain. And that's a large story that will be the subject of the next lecture.